So I hope everyone enjoyed the presentation that we had today by Roy van der Meel. And if you have any questions, feel free to just put them either in the chat or just unmute yourself already. We can do that at any time. For now, I don't know if there's anyone already having a question that they want to have answered straight away. If not, then I have a couple prepared. Is there someone who wants to ask a question? Yes, Mr. Wright.
Hi, this is Jay Wright, cardiologist from Liverpool, UK. Professor van der Meel, thank you very much for a great presentation. I really enjoyed that. We have a debate raging in the UK at the moment about the importance of the second dose, particularly with the Pfizer and Mrna vaccines. I'm fortunate enough to have had the Pfizer vaccine as a frontline cardiologist, but in the UK, we're now considering waiting 12 weeks for the second dose. So I realise this is a very clinical question, but I'd like to base it in basic science. How important is the second dose and how important is the timing?
Good to see you and thanks for the question. You probably know this better than I do. Me not being an immunologist, but a biomedical engineer. You know, we wouldn't have this discussion if they were able to produce enough of these vaccines. So maybe that already says something. Of course, you know, these trials that I was talking about, where they shown this efficacy, and maybe you saw it in the graph, is based on these two doses at a certain time after each other. And yes, there's now research coming out that shows already after the first dose, there is some level of protection. What worries me are the reports that I read from you know, concerned virologists that are saying, look, what you don't want to do is create an environment where you have, in some people, maybe a limited amount of protection. You create an environment where the virus can adjust, and you know, us, the whole world, calling the British variant a mutated version of the virus that is able to spread faster. Some people are concerned that if you don't stick to this schedule that they have tested, and the second dose, you administer that after a later time point, that you give the virus a chance, or at least in some cases, to adapt, and that could be a bad thing. I also understand the other side of it, and the other side being, you know, if there is a level of protection, can we not use that whole big batch of second doses to vaccinate more people? And as a Dutch guy with a government that has severely screwed up the vaccination regime in the Netherlands, with only less than 1% of the population being vaccinated, I understand that people are looking into this, but I cannot give you a definitive answer saying I would do this or I would do that. I can only look at what has been tested and what works. So I would, you know, go the safe way and say, do that. What do you think?
I'm very concerned that we are, as a country, making decisions on mass population health without following the science. Like you, I feel that the testing, the proof is in the 3-week second dose. There seems to be some emerging data from the AstraZeneca group that maybe, because it's working in a different way, not the RNA towards the spike protein, that it might be safer to leave for longer. But it worries me greatly. And I'm petitioning our hospital, along with the Liverpool School of Tropical Medicine, and the government, to try and bring this forward. Even if it's not 3 weeks to 6 weeks, they're proposing 12 at the moment. And I can assure you our government screwed up many more things than yours has in the last few years.
I'm glad we're on the same page on that. Thanks, Jay.
My pleasure.
I have a question here from Brian. He's asking, can the mRNA be used to treat very sick people as well as prevent infection?
So you mean vaccine-wise or mRNA therapeutics in general? I see Brian there.
Yeah, hi there. Yeah, so yeah, I understand prevention, but we have lots of people dying in hospitals. Could any of this technology be used to help those people who are very, very sick?
Very sick.
Already infected?
Difficult. I mean, mRNA therapeutics can of course be used to treat people that have, for example, a certain disorder where they don't make a certain protein, or they don't, or they make a wrong copy of the protein. For example, people that have certain disorders that they cannot make certain anti-coagulation factors. Those people you can help. People that are now in the hospital on the ICU. Vaccinating them will not bring relief to their situation. They first would have to recover from their current situation. And obviously, if you have had the disease and you come out of it, you of course have built an immune response against it. So for severely ill coronavirus patients on the ICU, maybe not, but in general, if you think about the rapid development that you can achieve with mRNA therapeutics in general, I mentioned the 6 weeks changing of the vaccine before. I foresee, at least in the future, because there will be a next pandemic, but there will be also a lot of other applications where these mRNA therapeutics can be very valuable and useful. I hope that answers your question somewhat.
Yep. That's great. Thank you very much indeed.
Thanks, Brian.
One of the reasons why people are not confident to take mRNA is that it creates an autoimmune reaction, which is not usually the case with other vaccines. How can we be sure that this won't create an unexpected result in the long term, the time that is longer than experimental phase? Are there any clinical evidences, such as cases showing impacts on long term?
Yeah, it's an excellent question, and this is exactly the reason why we thought to host this talk. These concerns about this rapid development and the long-term effects. So there's a few things going on. Yes, mRNA. If you put any type of nucleic acid, so DNA or mRNA, in your body, humans have developed over a long time and your immune system in particular, to sort out which of the nucleic acids belong to yourself and which do not, right? Everywhere in your body, there's all sorts of types of sensors. So all alarm bells go off when these things enter your body. So therefore, it also took such a long time to really develop mRNA as a therapeutic. If you look up Kathleen Carrico, the person who made groundbreaking discoveries about how to modify this mRNA so it is less, it still forms a template for the spike protein, but the mRNA itself doesn't cause an immune response. These types of modifications have really enabled mRNA to be used as a therapeutic. If you take a regular synthesized mRNA and you put that in a human or in an animal, you get a severe immune response. It's really these modifications of the mRNA together with, let's say, the delivery platform, so that you only deliver it at the right place, that make it so we're able to use it on such a scale. So the other part of your question about the long-term effects, the first people that have been vaccinated or that have been tested with LNP mRNA systems, there were, of course, other tests being done before the coronavirus outbreak. There's mRNA vaccines have been developed against Zika virus, Ebola, influenza, HIV, and they've been tested on more or less levels, preclinical to clinical. So I mean, from mouse to non-human primates to human. But the people that have been specifically the first people that have been treated with this mRNA vaccine against the coronavirus were of course the people in the phase 1, phase 2 trial. So we're now sort of a year down the line, and that's probably the longest term effects that they can really measure. As I mentioned in the presentation, it is not likely that there will be severe long-term effects. So mRNA, if you go to that blog post, which I mentioned in the presentation, which is about unraveling the mRNA code that's in the Pfizer vaccine, you'll see that the end of the mRNA has a poly A tail. And please go to that blog to find out what that is. But that poly A tail, every time that mRNA gets translated into the spike protein in the cell, a piece of that poly A tail comes off. So the cell can use this mRNA a couple of times to produce a spike protein, after which it is no longer useful and the cell gets rid of it. And this is a process that takes place in days. So within days, your mRNA is out of your body, your phospholipids and the cholesterol that are in the particle are being broken down by your body. There's only the polyethylene glycol. And so the side effects that you now see with the RNA vaccines are short term and they're very well treatable. Most people that get an allergic reaction, they have other allergies and they, for example, need to be treated with an EpiPen if they get a reaction on the spot after vaccination. I hope that answers your question a little bit. Of course, I cannot predict what is going to happen in the next 5, 10, 20 years. But it looks to be extremely safe. All I can tell you is that if I could get my hands on some BioNTech or Mrna vaccine, I would vaccinate myself, my wife, and also my 3-month-old twins if they would need a vaccination. Obviously, they're too little to need that.
I think that answers the question quite well. We have some other questions here in the chat. Can you briefly explain the process of identifying the relevant protein on the viral envelope and how the correspondent mRNA is found?
Yeah, that's also a very good question. Of course, I briefly skipped over that, and that was just something that was done really fast. Of course, this was done really fast. In general, there's of course a lot of work already being done on coronaviruses. We had the SARS outbreak years ago, then we had the MERS outbreak years ago. So from that research, people knew which proteins were important in, let's say, provoking an immune response, but also for getting into the cell. Now, to go from which protein is important to what is the genetic code. The genetic code of a virus is not that big. So we have large databases online where you can compare sequences and look at that code and see what that code, if you translate that into a protein, what happens. So from there they could see, okay, we have this part in the viral RNA. So first they identified, okay, the spike protein is important because that is the protein on the surface that gains entry and is also the protein that is being recognized by the immune system. Okay, second step. In the viral RNA, determine which part encodes for that protein. And then the third part, and which is very important, the mRNA for that protein, for the spike protein, if you put that into a particle, the cells will make the spike protein, but they make it in the wrong conformation. They make it in a conformation that doesn't look like, let's say, the active conformation. So it still shuttles to the cell surface. but it doesn't get recognized that well. So they had to actually do two mutations to make sure that once the protein is produced, it is put on the cell surface in the right conformation so it can be recognized by the immune system. Ernst, I know you want a more sort of a technical description probably of how that really works, and I'm happy to discuss it with you next time, because of course there's in addition to the let's say the lipid nanoparticle technology and the mRNA modifications, there's of course a third part which I didn't discuss, and it's the development of sequencing technology and also DNA printers and mRNA in vitro transcription. So I'm happy to talk, hopefully once we get vaccinated and in person. But thanks for the question.
We can do another talk on that.
I just wanted to say, you just give another innovation cafe on this.
I'm happy to do so.
All right, I have another question here. What type of immune cells are activated by the mRNA vaccines? Antibody / oh, sorry if I'm butchering this, cytotoxic T cells. Usually the body will know what type of immune response is needed based on the cytokines at the site of infection. How would that work for an mRNA vaccine when there's no actual virus?
That's again an excellent question. I'm really amazed by the level of the science here, and I also have to say that I'm not an immunologist, and that's why I also, to make things not too complicated, I didn't go into the specifics of the immune response of these vaccines. So basically two things happen, right? You want to create an immune memory, and you want to create an immune response that gets rid of the infected cells, let's say. And it's true that if you have sort of an inactivated virus that sort of mimics the viral infection, then the cell will process that as a normal virus and launch an immune response very comparable to when you get infected with the actual virus. That's why we've been using inactivated viruses for 200 years. In terms of when you, for example, use a protein-based vaccine, you really need to add something like an adjuvant. And an adjuvant is something that boosts the immune system to create a more strong response to get rid of, infected cells, but also to create memory and to create antibodies. So with the nice thing, I guess the one of the big advantages of using mRNA is mRNA by itself, as we talked about before in another question, in itself is slightly immunogenic. That means that by you can actually tweak and modify this mRNA code to make this immune response to steer it in a certain direction, let's say, or to make it heavier or stronger or weaker. But in general, these mRNA vaccines and the production of the spike protein on cells, or even if you inject these vaccines in the muscle, they will also reach sort of these antigen presenting cells already in the skin and in the muscle, and process, let's say, the mRNA and produce produce a spike protein or peptides of it, will likely give both a T cell response, but also induce the production of antibodies. And of course, that also leads to memory. So the mRNA vaccine is definitely able to launch an immune response, not the same as a virus, as you rightfully indicate, but strong enough to be protected when you get infected, that there is a strong response against the infection. I hope that answers your question more or less.
Dagmar, let us know if that answered your question. I have a question as well, because of course now we know now there's different variations coming up, like the UK version or the South African version. Can you say anything about how that does influence the vaccines that are currently there?
Yeah, it's also a really good question, and that was of course the biggest how do you say it? The biggest worry is that you make these vaccines and then the virus mutates. Vaccines don't work. I mean, this is of course the case why we don't have a vaccine against HIV, or one of the reasons. It's the reason why we have to make a flu shot every year, because mutations. And now the mutations that you see, this is the virus trying to change in a way that it's more advantageous for the virus. So that can be to spread faster or other advantages. A virus needs a host. The virus is not there to kill you. The virus is there to spread as much as it can, but it needs a host. Now, the reports that I've seen so far is that both the Mrna and the BioNTech vaccine are also working against the few variants that are out there, the Brazilian, the South African, the British variant. And it really depends how, let's say, how big the mutations are going to be that the virus will have. Of course, because you take an mRNA against the entire spike protein, if a little bit of the spike protein changes, the immune response and the immune memory of your body should still be good enough to recognize also that slightly changed spike protein. If it changes a lot, it might be necessary to develop a new vaccine. And that's why I also mentioned the strength of mRNA from code to cure. As Ugur Sahin, the CEO of BioNTech, mentioned, if that is the case, we would be able to change the code and produce vaccines with a different, let's say with the mutated spike protein mRNA within the course of 6 weeks. So when you think of a situation that this might be a thing that will be, let's say, occurring every year and turning into sort of a flu shot type of vaccination, the flexibility and the speed of mRNA vaccine development will likely be a massive help and very valuable there.
All right. Also, if there's any more questions, there's I think still a little bit of time, so please put them in the chat or you can ask them. In the meantime, I do want to ask, because you mentioned that there's like you showed us the graphs of like how the vaccines are tested. Are working, sorry. And you mentioned that there's like an over 90% efficiency rate basically of it working. And I was wondering, like, how can they test whether or not it works? Like, is it just like levels? Because I'm assuming that they're not exposing people to the actual virus. So can you explain? Maybe you already mentioned it a little bit, but
no, it is a good question, and in general, that would be a very good control group where you expose people to the virus. And there are actually trials going on where people act as volunteers and get and have themselves exposed to the virus to develop new vaccines or drugs. But what happened in these cases for the development of these vaccines, they asked volunteers to come in and get a shot. They didn't know if it contains the vaccine, yes or no. And after that, they just followed them over time and told them to just get on with their lives. Which meant that the people that had gotten the vaccine, and you saw that in the graph, they had a very small chance of getting ill. And where people that got the placebo, so the shot with nothing in there, obviously a lot of them got ill, but also in both groups, you know, there were also, of course, people in the group that got the placebo that didn't get ill. It's just the relative difference between those two that gave this sort of 90% efficacy. And of course, the 90% efficacy is based on these trials with thousands of people. If we you know, start vaccinating the entire world now, will it also reach 95%? I really hope so, but you know, that remains to be seen. But even, you know, even if it ends up working in 70 or 80% of people, the sort of the group immunity that we're trying to achieve by using these vaccines, that would already you know, hopefully bring us back to normalcy somewhat.
I mean, because what you're saying, like these people are being basically sent to go back to their normal life, but at the moment for nobody really the life is normal, as in there's still social distancing. So I'm really curious to see what will happen. I mean, that's just I don't think there's any other way than to see what's going to happen when everyone is vaccinated.
Sure.
But
and that's why these numbers also need to be that large. You know, you can also ask this for 10 people to do this, but you might not even see an effect, and that's why they need so many people to test this on. Also, of course, to make sure not only that it works, but that it's safe.
Another question from our audience: the approach that is being used to produce the vaccine, is it repeatable with ease for any kind of mRNA virus in case something else pops up in the future?
So the approach to produce the vaccine, you mean like the technical way that the vaccine is produced is very easy. As I said, if there's something else and they can change the code, you put a different mRNA on the inside. The way these things are produced is that you have this, the components of the lipid nanoparticle is dissolved in an organic solvent, let's say alcohol, and the RNA is in a buffer. And basically they have a massive, these two streams, they collide in a massive stainless steel, let's say T-junction. And those particles, and that's the beauty of the science developed by Peter Cullis, who I mentioned, where I spent some time in his lab, is that based on charge interactions and other interactions, these particles self-assemble into roughly 80 nanometer nanoparticles. And because they have that little PEG corona on the outside, which is hydrophilic, it makes sure that the particles, once they are produced and they are in solution, they don't let's say aggregate or clump together. They stay as these virus particles. So is this approach usable for every virus? That is really depending on how fast, you know, a virus is of course mutating, but in general, yes, you could of course, when there's a next pandemic, I think, you know, this approach can certainly be used, or at least try and see if that works to develop vaccines against the next big thing, which is surely something will come.
Great. If there's anyone who has a last question, there is some time left. So I'm gonna, oh, there you go. There's another question. If you are about to get a vaccine shot, is there anything you can do to boost your immune system to help the vaccine function better in your body? For example, make sure you sleep enough the days before.
That's quite an interesting question, and I don't think I really have a good answer for that, other than it, of course, you know, if your immune system is working properly, if you're healthy, you have to imagine that every day we get exposed to, if you go outside, which we don't do so much anymore, but normally your body gets exposed to maybe millions of invading viruses and bacteria, and it's super capable of fighting that off. But you also know that if you're, you know, if you're not been treating yourself well, in terms of maybe diet or maybe exercise or just, being healthy in general, your immune system also suffers. And you can get, you're maybe more susceptible to catch a cold or to catch the flu, or maybe get a cold sore or whatever. So I think it definitely helps that, if you're in a good condition, your immune system will definitely be able to work properly, and that can help. But that's sort of more of a general thing about vaccination. It's not like if, for some reason, I've also gained a few corona kilos and having exercises regularly with the twins at home. That doesn't mean that the vaccine doesn't work. The whole reason to get these 2 shots is to really boost the effect of the first shot. So it definitely helps if you're healthy, but if you're not so healthy, don't worry, let's say. I don't think that's the reason why you asked the question, but yeah.
Great. I think that was enough. Well, it wasn't enough. I think we could keep talking about this for a very long time, as we have for the past, year already. But we are running out of time. So hereby, I want to thank everyone for the questions, and I want to thank Roy a lot for answering all of the questions and the great presentation.
My pleasure. Thanks for having me.
We hope to be able to join, to have you join another time to maybe go into more details. But for now, that was it for today. For next week, we have another innovation cafe. If you want to see, it's organized by Art and Tech Society. And if you want to see what it's about, you can go to our website, ehvinnovationcafe.org. And also the talk from Roy will be posted there, and it will stay on YouTube, as well as this Q&A will be posted there as well. So you can rewatch as much as you want. And for now, I just want to say thank you for joining and have a good evening.
Thanks, everyone. Thanks for joining.